or really, I'm not going to really present anything clinical, but I'll try to allude, I'll try to vaguely allude to it as much as possible. So feel free to ask questions about implications for structure and clinical function. So just beginning, for what I study and what I study with the behavioral function of the default mode network, there's a lot of processing that we don't commonly study in psychology, mainly making novel decisions very rapidly, things we have to do every day, but without much learning. For instance, yesterday I was leaving my office in central London that I was visiting for a while, and I had to get down here within an hour, check into a hotel in Camberwell, another place I've never been. I haven't ventured to the depths of South London, and also meet a friend for dinner right afterwards in another place I haven't been to. And being able to deliberate about one choice is difficult enough, but we also have to plan sequences of choices and look ahead into the future and use past experience in order to guide our novel decisions. And this is thought to rely on an internal model of the world, because I have to explore what I know about, from my experience living in London, how long it takes to get to the depths of South London, how much I need to walk, whether I want to carry my luggage around to get there. And all this past experience can help us inform our novel decisions. And that's been a new perspective of looking at the regions in the default network, and mainly not looking at stimulus and response, but internally guided cognition, being able to generalize from past experience without having it be our present decision or present state, being able to look beyond our present state. So just a brief history of the default mode network, just so, because I know there's a variety of backgrounds here, and feel free to stop me if anything's unclear, because I don't want to rush over some complicated concepts. But early human neuroimaging studies with PET and fMRI, they were consistently observing brain regions that were more active during baseline periods than the actual tasks. And they were calling these task-induced deactivations. And no one really knew what it was, and people just took it as like, oh, these are the baseline regions, even though many influential people thought the idea of a baseline in neuroimaging was not useful in itself. Then around the mid-90s, a psychiatrist, Nancy Andreasen and colleagues, explored the importance of spontaneous or internally directed cognition in these like internally guided behaviors. So behaviors like imagination or autobiographical memory, things of which wasn't really commonly looked at with PET or fMRI imaging. Most people were doing working memory tasks or something where there's explicit a stimulus, where you're looking at some visual thing and you have to react to a decision. So things that are ongoing processes that we need to generate ourselves instead of reacting to what's being presented to us. And it was, and this resting activity in these regions was thought to correspond to this, particularly these parietal and frontal midline regions. Then shortly thereafter, researchers at Washington University in St. Louis, in the lab of Mark Rakel in the late 90s and early 2000s, compiled meta-analyses of regions that were always commonly deactivated during their attention tasks or during their working memory tasks. And he referred to this as the default mode network, assuming that this is the baseline mode of cognition. When we're not doing anything, this is what the brain does. But that in itself was very unclear. The regions that were part of this default network were medial prefrontal regions, like both ventral and dorsal medial prefrontal cortex, which I'll discuss in detail later, inferior parietal lobule, and the parietal midline, posterior cingulate and retrosplenic cortex, and the hippocampal formation also. And they thought they would function like a visual or motor network, but instead just be directly corresponded to internal mentation, or imagining the future, in other words, or self-referential behavior, things that related to our subjective feelings or subjective states. And eventually it was found, this may have seemed trivial at first, but they first referred to it as a network just because they were commonly deactivated at the same time. Then a couple of years later, researchers at Stanford found that also the signals between these regions were correlated. And this was when resting state networks were popularized about 10 years ago. So even though frontal and parietal regions are very distant, they're correlated with each other. And later, structural imaging also showed that these default network regions are highly interconnected. And now there's hundreds of papers every year examining the architecture of the default network, differences between clinical populations, and anatomical observations across species and parcellated subnetworks. But the problem is, we know what these regions do during behavior when you have a certain task, but really what is the behavioral or cognitive significance of spontaneous activity in these regions? Why are they so fluctuate? Why are the amplitudes changing and so tightly correlated when we're doing apparently nothing? A clue might relate to the behaviors that they relate to when we're actually running tasks. So tasks related to episodic memory or planning the future or evaluating choices in mental imagery also elicit activation in these same regions. But the behavioral relevance of the fluctuations in these regions when you're not presenting a task is unclear. In essence, it's not the default mode. It's not just a baseline of wild mind wandering. But some clues might come from the strength of these resting correlations. So looking at the correlations, how tightly correlated a frontal region is with a parietal region, for instance, and see if that correlates, the strength of those correlations correlates with some behavioral variable across individuals. And some studies, to a varying degree of success, have used those correlations with individuals learning something and then having a 10-minute resting scan and then being tested on what they just learned and seeing if those correlations predict memory performance, for instance, or the retention of what they learned. And there have been some studies that successfully looked at it. So it's putatively thought to correspond to memory consolidation. But an even more interesting hypothesis about resting state networks that could come from neurophysiology and studies in animals. There's a recent review in Nature Reviews Neuroscience by Ken Harris and colleagues, is that spontaneous neural firing during rest seems to reflect past behavior and possibly encode prospective behavior. So what you're seeing here is behavior in a mouse, and the neurons that fire in a mouse when they're listening to a tone. And there's a specific sequence in the different neurons fire. But then when you look during rest, the spontaneous firing of the neurons occur in the exact same sequence. And any deviations can predict which neurons are going to respond when there's a novel behavior. So you can sort of think of this as a barcode or a serial code, that maybe spontaneous activity is assigning which neurons are going to fire when there's a novel stimulus presented in advance. So it's not that they're predicting the future, but they're assigning which neurons are ready to fire. And this could be an organizing brain principle. And if we think of this at the level of resting fluctuations for fMRI or whatever signal we're looking at, maybe those fluctuations can also reflect how the brain is going to respond to upcoming choices and perhaps past experience. And if we think of this in the default network, since it's using these regions that have high metabolic demands and really behaviorally demanding things like imagination, which utilizes our memory, our preferences, decision-making processes, and vivid sensory information, that would give it an excuse for it to be the most prominent resting state network. In other words, it could be internally reflect the internal exploration of previous experience in order to prepare us for the future. And one of these sub-regions that's linked with the default network is the hippocampus. And I think everybody here is familiar with the hippocampus, the human hippocampus is up there. What people might not be as familiar with is in rodent literature, pretty much right after John O'Keefe, Nobel Prize winner, discovered the place cell, the cells in the hippocampus that correspond to a specific location, that there was spontaneous high-frequency firing around 80 to 120 hertz when the rat was doing nothing, or when the rat, and especially when the rat was sleeping. And there was these, you can see it there, there's a little black squiggle. It's during these restful deliberative periods, there are large deflections or sharp waves in the hippocampal local field potential that co-occur with these oscillations, and they're very high in amplitude generally. There's other electrophysiological events in the hippocampus, but this was the most prominent. And interestingly enough, these ripples are associated with memory consolidation, since these reactivation of hippocampal place cells ensembles, so sequences of place cells, or which you can see as sequences of locations, in which rodent literature, people studying rodents, that's what they'd like to associate with, like an episodic memory, that a sequence of locations could be the architecture for an episode that we remember, or a context that we remember, for instance. That this could be the sequence of place cells that fire during sharp wave ripples, could correspond to the reactivation of a memory, perhaps in order to strengthen a memory, or to, if you could think of it in psychology terms, sort of a mental exploration or transitive inference. So if I activate one sequence of one location with a sequence of other location, I can integrate that information to create an even bigger cognitive map and explore sequences that I haven't explored before, and be able to link these two contexts together and maybe generalize for future exploration, which is a quite interesting idea, I think. And in further support of this hypothesis, the number of ripples after learning predicts subsequent memory performance in rodents and also humans, because it's extremely difficult, if not quite impossible to, I mean, it's been, similar things have been done in humans, but in epileptic patients who have electrodes in the hippocampus, you can definitely measure these 80 to 100 hertz oscillations of ripples. I mean, if not directly in measuring the actual cell firing that co-occurs with it. So the proxy of measuring those ripples when participants were taking a nap, if they did a memory task or learned memory associations before a task, took a nap, and they measured ripples during the nap in the hippocampus, the amount of ripples predicted their performance. That was a brain paper from about 8 years ago from the epilepsy group in Bonn. And even interesting and more causal mechanistic role is that electrical stimulation of the hippocampus after learning interferes with memory consolidation. So in a series of clever studies, people studying ripples in rats, after rats were learning reward locations in a maze, they electrically stimulated the hippocampus when they observed a ripple, and this interfered with their memory performance. And even more interest to a systemic level or a default network level, because it's very difficult to measure multiple areas outside the hippocampus in rats when you're measuring ripples there. Nikos Logothetis' lab in Tübingen developed a technique called neural event triggered fMRI. So what they do there is they have electrodes placed in the hippocampus in order of macaques, in order to measure ripples or resting activity in the macaque while they're anesthetized, but also at the same time have them in an fMRI scanner to be able to capture fMRI, the fMRI signal across the whole brain. So there they could link the onset of specific events in the hippocampus, like low frequency oscillations or gamma oscillations or ripples, and see the bold changes that co-occur with that. And what they found excuse me, is that voxels across the neocortex and deactivations in subcortical regions, specifically after ripples, but not these other hippocampal events occurred with these neocortical activations and subcortical deactivations. Thank you. And as you can see, if you look at the ROIs of these regions, you see these transient increases in voxels across the whole brain, just after these very local events at the level of hippocampal circuits. So just a series of neurons with a really high amplitude firing can change voxels on the other side of the brain. But what was unclear was whether this was in training a whole network, or is this really high amplitude fluctuation, high amplitude activity propagates to the rest of the brain, and this activity decays. And luckily, in collaboration with one of my supervisors in Barcelona, Gustavo Deco, we were able to analyze this data and look at whether this was also changing ongoing networks, and that the point made in Nikos's original Nature paper of that there's subcortical deactivation and neocortical activation, that the whole cortex was awake to be able to receive propagating activity. If there's something more specific, if these resting state networks, like the default network, which you would think would be modulated by these very events, and perhaps not other networks. So the questions we had when we had access to the data are hippocampal sharp waves influencing ongoing resting state fMRI activity in unitary resting state networks, or is it just diffuse transient activation across different neocortical areas? One clue that was that the areas that showed some of the greatest amplitude changes were default mode network regions. But it's still not trivial whether it was affecting this ongoing activity, or it was just these closest areas had a huge transient increase because they were structurally the closest areas to the brain, or a mixture of both. And are they influencing some resting state networks, like the default mode network, more than others? And this would be an opportunity to potentially relate behaviorally relevant circuit level activity, so just what I was saying in one of the earlier slides of spontaneous neural firing related to behavior, to actual spontaneous brain network fluctuations, which no one's really done. So hopefully, I was hoping we could provide very preliminary evidence that resting state default network fluctuations could or at least generate a mechanism to show that mental exploration of past experience by these reactivations of priorly visited locations or prior learned representations might be reactivated in order to prepare the default mode network for the future. This would be a first aim of doing. So what we did was, across many sessions in 2 macaques presented in that Nature paper, there's 25 sessions of 10 minutes each in each monkey, so we had thousands of ripples and other neural events to regress against the resting state signal. We used a technique in collaboration with Dante Mantini to align the resting state networks that were present across both monkeys and across all sessions. And then, and so these are just examples of each monkey. So this is the first monkey and the second monkey. And this is just a typical representative default mode network signal. So just the signal of all those regions collectively. So not just one voxel from that, but the whole thing. And these red dots are where ripples appear. And essentially we're just running a correlation between this, but also the onset of other events. And as you can see, there's ongoing activity. So what people in animal physiology don't really study resting state don't realize is whether they were just thinking that maybe 5 seconds after the onset of the ripples, you'd see an increase in signal across the network, and then it would just decay again. But in fact, the important thing here is that it's an ongoing network, that there's slow waxing and waning of the signal, and that potentially these ripples were slightly increasing that waxing and waning, so more of like an entrainment process on this. And as a control, to give more specificity in the neocortex, because even if we showed that ripples selectively affected these the default mode network more than gamma or sigma oscillations, we couldn't be sure that it wasn't just the whole neocortex doing that. So we used the most robust network we observed, the one that was strongest correlated across monkeys and across the sessions, using this independent component analysis technique. And that also wasn't linked with episodic memory consolidation, so the ventral somatomotor network. And what we observed, as you can see across both monkeys, is that after ripples, there was a huge increase in default mode network activity, but not in the ventral somatomotor network. And these increases were specific just to ripples. And we looked in other networks. The difficulty of this was there was not as many robustly observed networks beyond the default mode network in the ventral somatomotor network in these monkeys that were present in all sessions. But in some of these more robust networks of primary visual cortex or occipital-temporal cortex or frontal-parietal attention network that were present in at least half the sessions of the monkey, we also saw that the default mode network fluctuations after ripples were much stronger. So what can we conclude from this in the interim? We find that hippocampal ripples, but not other events, correlate with increases in ongoing default mode network activity. The default network is the only resting state network displaying consistent signal increases after hippocampal ripples. I'll allude to this in one of these next points, is that also these cognitively relevant spontaneous neural firing, so just at the level of groups of neurons, are influencing ongoing resting state fluctuations in the fMRI signal. And importantly, and I think this is an aspect that's ongoing in our research in Barcelona and of interest to people studying neuroanatomy, is the likely mechanism for this is that the hippocampal ripples have an extremely high amplitude, an order of magnitude higher than the other events that we're showing. And given the tight anatomical distance and connectivity between the hippocampus and retrosplenial cortex here, posterior cingulate, medial prefrontal cortex, it's likely the primary target for propagating output from the hippocampus. So that's not to say that these regions are the default network's the only thing being affected by hippocampal ripples, but it's to say that it's the primary target. And then this resting state default network activity could possibly relate to mental exploration of past experience, although that needs to be modeled with actually looking at place cells. So now we've looked at resting state and kind of this functional architecture of what propagating neural activity can affect, that's behaviorally relevant, can affect a resting state network. But what about the network itself? We mentioned all about future decisions and future choices, but it's really unclear how we use an internal model of the world to generalize and make novel decisions. Because usually in a task you have to train participants or even overtrain them in order to, and that's why reinforcement learning, especially in the department I did my PhD in, is so popular, because you can control exactly what they're learning. But what about these rapid inferences we make without any learning or little learning? Because we have the capacity to quickly infer these future outcomes even with little or no learning. And this is generally associated with higher reasoning or abstract reasoning in dorsal, medial, and lateral prefrontal regions. The most evolutionarily advanced regions, I guess, as some would say. But teasing apart the respective contributions of these medial PFC regions involved in a default network and these advanced lateral PFC regions like dorsolateral PFC or frontal polar cortex has been difficult. And the everyday ability to effectively plan with little or no learning is unclear. Like, for instance, choosing where to proceed when you're at an unfamiliar intersection in order to go home. It's been observed, and this is interesting and hopefully maybe of interest to psychiatrists, is dorsal portions of rostral MPFC, particularly since Chris Frith studied and Uda studied internal mentalizing, so inferring the beliefs of others, that actually might relate to a more general contribution to making inferences about future states. So making inferences about a given decision in the moment that we're going to execute at the moment, or versus modeling a decision about a decision we're going to make, but not at a given moment. So at a different subjective state. Be able to move beyond a subjective state and save a choice for later. So how we looked at this is we adapted a task that would elicit ripples in rodents. So like looking at a T-maze in rodents, commonly when a rodent is awake, usually ripples are observed when rodents are sleeping. But also when a rodent is at a choice point in a maze, so when they're at a T, when they're deciding which direction to go in, or deliberating, as some people would say, about where a reward is. You see all this spontaneous firing and replaying of prior learned representations and of the location, those upcoming locations. So we adapted a similar task, and we were under the assumption that goal-directed exploration of real and mental space uses the same internal model based on our exploration of the physical world. So that's essentially saying, put simply, that's when we're mentally exploring, so when we're thinking about our commute home, we're using the same brain regions and making similar contributions when we're actually moving or on our commute home. And using such an internal model, mental exploration would allow participants to make rapid spatial inferences with little learning. So we wouldn't need to train them on these abstract spaces. They just have to choose what the shortest distance is when just viewing a novel maze. And specifically, we engaged participants' inference about the shortest route between a starting and goal location within a maze when they're shown an overhead viewpoint of it. Don't worry, I'll show you a task and I'll go slowly through it so you get a good idea of what participants were doing in this task and what I was interested in looking at. And to investigate prospection, so future choices, some mazes had a choice only at the starting point, so just an initial decision or a current decision, while others also had a choice point further in the maze, so sequential choices. So they'd have to remember the first direction they'd go that would be the shortest, and then they'd also have to remember another choice and have another choice point further in the maze. So then we could see whether there was a different computation going on for the present choice versus the choice that they were going to have to make after that present choice. It will be clear when I show you. And so after viewing a maze for 3 seconds, participants would then need to choose the correct direction for one of those choices. But they didn't know if there was two choices. If there's one choice, they'd obviously know which choice they would need to make. But if there was two, they'd have to maintain both of those choices. And so they're presented with a maze like this. And so this would be a maze with a sequence of choices. So we'd technically have 3 choice points, but we'd only test them on choices along the optimal route. So they'd have to choose left. Within 3 seconds, they'd have to choose left or forward here, and then also at the second choice point. And then after 3 seconds, it would be highlighted, and so they know they would have to make that choice, and then it would zoom in on that point, and they'd have 1.5 seconds to make a decision. And from that, we could gather the reaction time to make that decision and gather their uncertainty, because after a while, they were getting pretty good at this. So it would be all novel mazes. So these would be things they'd never seen before, but obviously they had a knowledge of space, so they could judge distances very rapidly. So here would be one with only a single choice point. And so they'd obviously know within these 3 seconds whether they wanted to go left or forward, but I mean, it still could be difficult. And they had mazes of varying difficulty, of easy, medium, or hard mazes, and I'll kind of explain that a bit better on how they do that. So the regressors, or the factors that we were looking at in this experiment, were for mazes with only one choice, it would be the difference in the path length from the optimal route, or the shortest route, versus the other option. And that would be how difficult it would be. So it would be similar to how people look at decision making, if people study decision making, the difference between an optimal value and a suboptimal value, or a value difference, which is already known to engage similar regions in the prefrontal cortex and parietal areas. Or for the four routes, you get the distances of all the choices and the differences between all of these 4. and that would correspond to their uncertainty about that. But also, we could just focus on prospective choices, so be able to separate the amount of choices versus where in the sequence they needed to make a choice. So this was only about the difficulty of the two options at the second choice point, regardless if they needed to choose it or not. So the second choice point of the two choice points in the sequence. And to further get a variable of the participant's uncertainty, we also, and I can explain in more detail later, but I'm sure most people aren't interested. We used the reaction time during the decision phase to get an idea of their particular uncertainty, because participants were performing close to ceiling, or at least we made sure they did, because we were more interested in the differences between mazes than the differences between individuals. And so these are the three contrasts I'll show you, but mainly we'll collapse across these two and just look at choice uncertainty and then more prospective decisions. And just to give you an idea of just these differences in values, so this would be a one choice maze that would be a bit easier. Obviously, we know to go right. And this one would be a bit harder. And then here's a maze with two choices where the initial choice is harder, and the second choice is a bit more easier. Whereas this one, the initial choice is, oh sorry, I mixed it up. The initial choice is easier and the second choice is harder. But they still needed to remember these regardless. But that way we can dissociate between initial difficult decisions versus future difficult decisions. And if it's just the same general process or not. And so just overall the choice uncertainty without this prior learning, no matter how many choice points were in the maze, we observed what you would see with a value-guided choice task, of whether you have these huge value differences in the peoples in the ventral medial prefrontal cortex. So like the easier it was, or the more certain they were, it would be ventral medial prefrontal cortex. And then pre-SMA, dorsal ACTC sort of attention areas were engaged when they were uncertain about an initial choice. Interestingly, kind of hinting at that these more rostral areas of dorsal MPFCs, more theory of mind regions, that's usually what these areas are associated with. Not that we have any proof of that. Were engaged when there was more choice points, when there was multiple choice points in the maze, for decreasing choice uncertainty, when people were more certain about multiple choice points in the maze. Whereas these more ventral regions was when there was only one choice that needed to be made, the current state, and you needed to signal that preference, and they were certain about that current state. So either way, this is they're certain, but this is corresponding to being certain about many choices versus being certain about your initial choice. But that's not proof in itself of perspection mechanisms. So when we just looked at what correlated with the difference in paths just at that second choice point in trials that had that, what we observed was a dorsal MPFC region just caudal to the blob that we saw there, and just anterior to these dorsal ACC regions that selectively responded to increasing uncertainty about these prospective choices, but neither single choice, mazes with single choices, or all four mazes or the amount of choices. It was just to the second choice, uncertainty about the second choice point in a sequence of choices, or the last choice point in a sequence of choices. Whereas the other regions that we saw that corresponded to this prospective uncertainty were more generally responding to to difficult decisions overall, or the amount of choices, but were engaged even more when there was a prospective choice. Does that sort of make sense? Interestingly, and a bit more confusingly, and I'll make more speculation about this, is the other region that we saw that was kind of surprising was the dorsal posterior cingulate cortex. Dramatically changed its response depending on whether there was a difficult choice later in the sequence or an easy choice at the beginning. So here when we have an easy choice at the beginning and a difficult choice at the same, it changed its response. So we see the more certain they are for initial choices, the dorsal part of the posterior cingulate responds. But then the more uncertain they are about the future, it also responds. So it switches its response, whether it's certain or uncertain, depending on the choice sequence. So what this suggests is that it's responding to these shifts in the task. So when they're certain about something, they need to move to a different phase of the decision, so that choice phase where they're going to press a button about where to react to, or move to another choice point to make a decision. That's what this posterior cingulate is responding to. To disengage from this choice, because the choice is already known, the preference is already known, to focus on this more difficult choice. And this is quite interesting because it's known to respond to shifts in strategy to other phases of tasks, but also because of the anatomical projections through the anterior cingulate to this rostral MPFC region. So just to summarize, if my explanations didn't make that much sense to you, is that the VMPFC more corresponded to easy current choices, and then as you move dorsally, it was more easy future choices, and that these rostral parts of the VMPFC were difficult future choices. So you see the areas that are most heterogeneous across individuals also correspond to the more complex decisions, just with different forms of uncertainty. And then difficult current choices were pre-SMA, and dorsal ACC. So this would be the area of the midline area of Brodmann area 9. And this would be more Brodmann area 8. And this would be Brodmann area 10, and more ventral regions here. So this might be imply that there's differences from an anatomical perspective. I find this very interesting because this might imply that there's differences in recurrent connectivity within the medial wall. So obviously, this is likely due to the very different cytoarchitectonics between the dorsal ACC, pre-SMA, and these more rostral MPFC regions, where there's even much higher neuronal density here versus here, where it's much sparser. So you might think that for a longer sampling time that you need for relative, like maybe 2 or 3 seconds to make a decision versus 1 second, that would require more recurrent connections within these sparse regions versus diffuse, shorter time scales that you see here, but also see here. So it gets, someone feel free that knows more about this than me, that the neuronal density as you move up the medial wall gets sparser and sparser. And I think that's interesting, and that's also reflected in the response, because you see the dorsal ACC is much more distinct. There's a distinct decrease by prospection demands from a current to a future choice. Whereas here, the slope is generally the same, moving from a current to a future choice, but it's biased towards the more future choices in the dorsal, the more dorsal regions of rostral MPFC. So that would suggest that there's some sort of anatomical constraint here in a region that's all included in the default mode network and sometimes shown as a diffuse activation. So just the conclusions when I talk about, I'll also list some more speculation so we can discuss later. This spontaneous hippocampal circuit level activity selectively influences the default mode network and the fMRI fluctuations there. These same default mode network regions help make rapid inferences about future choices. And so that would suggest that these dorsal and frontal and parietal midline areas allow us to make rapid and robust inferences about a sequence of choices, which might also underlie our ability to impute the intentions of others. So making a inference about a state beyond our own current state might also be the same computation related to making an inference about someone else's state versus our own subjective opinion. And more speculatively, these rapid prospective inferences without prior experience can be computed from mapped knowledge, which would be these place cells in my example, but this could be more general across cortex. And mapped knowledge of space could be reflected in the form of hippocampal place cell ensembles. And this spontaneous reactivation of place cell ensembles selectively influences the endogenous fluctuations in the default mode network more than other resting state networks. And this also suggests that these different default mode network and cortical midline regions potentially have different levels of recurrent connectivity and anatomical heterogeneity in order to either make single or sequential choices. So this also might imply that rostral DMPFC regions and that their development and dysfunction might be key precursors to mental illnesses and degenerative diseases. And depending on the factors of this development, whether it's recurrent connections or anatomical heterogeneity connections with other brain regions, that might affect what computations they make and where the dysfunction would be. And that's a very interesting area for future computational modeling, to get the biophysical properties of a decision computation, whether something's biased towards a present decision or a future decision, and seeing if that depends on the connections with other brain regions or the recurrent connections within a brain region. I'd just like to thank my PhD supervisor, Neil Burgess, my current supervisors, Carl Friston and Gustavo Deco, and my other colleagues at the Universidad Pompeu Fabra and UCL, and also Dante Mantini for the ICA script, and of course, Nico Solifedis and Yusuke Moriyama for spending years collecting this really difficult data and letting me analyze it. And I'm, looks like, supported by the Wellcome Trust. But it's a welcome full-time fellowship. Thanks.
Thank you very much. Question?